Pardon me, sit. No. Okay, I'll get started.
Yep.
Okay, great. Thank you very much for inviting me today. I'm very excited to be here. My name is Matthew Ricci, and today I'm going to be talking to you about some actually very recent work. I made the controversial decision of talking about something that's sort of taken over my thought process and the thought process of my colleagues in the last couple of weeks. Still very early days, but something that we're really excited about. And I thought it'd be a great opportunity to have a discussion with you and hear your thoughts and learn your way of thinking about this. And what we're going to be talking about is finite dimensional representations of a particular type of partial differential equation, which finds particular expression in biology, namely reaction-diffusion equation. And we're going to be talking about what the meaning of these finite dimensional representations are, what they tell us about reaction diffusion processes from a physical perspective, and what that can in turn teach us about biology. And quite frankly, one of the reasons why we're so excited about this these days, this early work, is it was frankly so beautiful. Reaction diffusion processes underlie some of the most striking examples of spatial patterning in the biological world, but really the natural world. So what you're looking at here is a time-lapse video of a growing biofilm. Now, biofilm is a bacterial colony. 90% is estimated of bacterial colonies in the world exist in these collective formations. And here you see it growing in time. And without knowing anything about the underlying processes, you can see that there's a lot of interesting structure happening in both time and space. You have an obvious, interesting radial symmetry, where you get this multi-scale phenomenon where you have large structures coexisting with smaller ones and yet smaller ones, etc. And the theoretical import of the reaction diffusion mechanism is its manner of connecting microscopic physical processes to large-scale macroscopic qualitative behaviors in space and time that often manifest in these absolutely entrancing patterns across the biological world. But they're not just in biofilms, as exciting as biofilms are, but they're everywhere. And when I talk about reaction diffusion processes, I'm going to be having in mind a certain basic functional form of a partial differential equation. And what that PDE describes is the behavior of, let's say, an evolving concentration of chemical species, or just a diffusing substance or substances, which we'll call U. And as the name suggests, it's subjected to a diffusion process and also local interaction kinetics. And those two processes of diffusion and reaction are parameterized. So there's some diffusion constants and there's some reaction parameters. And collectively, we call those the moment theta. And often we'll just abbreviate the equation of motion to the action of some generally nonlinear operator n. And inside this unassuming looking mechanism lives a whole world of beautiful, interesting spatial and temporal structure that we see emerging across the biological world. And those beautiful patterns are caused by what's known as a diffusion-driven instability. And that's where an initially uniform, steady state, spatially homogeneous state is destabilized in the presence of diffusion. And under certain conditions on theta, that instability causes particular wave numbers to grow and then stabilize in a manner that reflects these gorgeous patterns. And it's not just theoretically interesting, it's also crucial for applied sciences. So it emerged originally from early work from the 50s on embryogenesis, the beginnings of human development, protein dynamics, which is something that I'm very interested in, and more synthetic biology. So actually building these things in the lab and manipulating them. And so I have a couple of examples here. We have maybe the most famous examples of animal pigmentation, which are supposed to develop by similar processes. In the biological world, it occurs across scale, so not just the level of cellular interaction, but also at the ecological scale. So you can see in the formation of plant life and forest arrangement, you get these spatial patterns controlled by similar mechanisms. Here I have an example of the fairy circle, which is a collection of fungi that grow in these spot patterns. People used to think that they were where fairies met at night and danced and played. And now we know it's actually even more amazing than that. It's actually reaction-diffusion dynamics.
Sorry,
outside of biology.
do you mind if we
Yeah.
interrupt you while you're going on? So Pierre has his hand raised. Is that okay?
Yeah, sorry, I couldn't see.
No, I'll call him. I'll go ahead, Pierre.
Yeah, I just wanted to ask about the previous example that you just showed, the biofilm. Do you claim that it's governed by reaction diffusion, such as the equation that you have?
Okay. So
because it looked a lot like wrinkling, which is something that you can observe in a purely mechanical system.
Yes, and I will admit to being persuaded mostly by my aesthetic sensibilities here, because this I would argue that while there might be some reaction diffusion processes here, which can be significant, there's also a lot of other things going on to them. And notably, there's like transport in the form of advection and things like that. But this is suffice to say just a striking example of pattern formation in biology. These, on the other hand, are really reaction diffusion processes subjected to the classical Turing story. But that's a very good point, and you caught me succumbing to my aesthetic instinct. We can talk more about biofilms in particular later. One question I'll ask at the outset is, should we do it talk questions or discussion? I sort of prepared it in the former way with the talk first, but if there's things that are really unclear, people are welcome to join. It's whatever we decide.
Yeah, let's leave the long questions for later and the short questions, just if somebody has some confusion.
Okay, yeah, that's perfect. Okay, great. Now, in general, it's hard to understand the way these things behave, especially when they're modeled in biophysical detail. And often biologists want to know things like, well, when do stripes appear and when do spots appear? When is it stationary and when is there some sort of chaotic behavior? In general, it's difficult to know when those macroscopic qualitative behaviors emerge in these things. And that is a significant bottleneck for the applied work. And so often what you'll see when people are working on these things in the lab is that they resort to parameter sweeps. So if they want to know in this evolving system of proteins, which I have on the bottom right from recent work by Glock, if they want to know when it's stripes or spots, they'll just simply sweep over all the parameters, which is costly in as much as it does not lie on targeted interventions supported by some sort of theory of what I'm calling suggestively qualitative dynamics. So it's everywhere, but there's already some interesting questions to ask. And the problem is that it's difficult to predict patterns. So if we want to know stripes versus spots or all the other beautiful things that arise in the presence of very strong linearities and sometimes couplings between spatial scales. That's sort of a nightmare scenario. So the classical work, or what you would maybe think to do first is you take the equation, you linearize it about a fixed point, and you see what it does. And we know now that that can tell you at least something, but not too much. So if you linearize, you'll see that under certain parameter conditions, there are often a range of unstable wave numbers that will grow in time. and others that will decay in time. And in general, this is the scenario according to this so-called dispersion relation, which relates the wave numbers to the growth rates, which tells you when patterns will appear, but really only what their scale is. And when Alan Turing, who actually began this work with his final paper in the early 50s, studied these things. This is sort of as far as he got. Later work resorted to perturbative methods, but in general, those can fail in the presence of very strong nonlinearities where your perturbative assumption does not hold. And then you can get esoteric cases where you have things like solitons, which can collide and in so doing disobey your perturbative assumptions. The most recent theoretical work I'm familiar with is in renormalization. And in general, that also sort of works in the same assumptions that the perturbative framework does, but it tends to struggle when there's scale coupling. And on the subject of renormalization, I'll say that one of the reasons we find this so theoretically interesting, again, this early work, last couple of weeks really, is encapsulated by a statement from a now seminal paper by Jean Bricmont and Antti Kupiainen studying reaction diffusion equations with renormalization. And they begin their paper in '94 with the statement, the development of a qualitative theory of infinite dimensional dynamical systems is a major scientific challenge, and one that we've seen now has practical effects for applied scientists making targeted interventions in these systems in the laboratory. And so we're very much intrigued by the pursuit of a qualitative theory as it emerges, or as it could be applied to these beautiful pattern generating systems in biology. And in general, this is my bread and butter. This is what I care about in my scientific work, because I'm interested in qualitative dynamics in biology. So I'm interested in things like equivalence classes of dynamical states in biology, asymptotics, regularity, heuristics. And I'm interested in pursuing those subjects using tools from physics, applied math, which is my background, and more data science, which is having a pervasive effect on the physical sciences. And we've pursued those qualitative aims across several areas. Some time ago, we had some work in complex networks and synchronization. With my current collaborators, we had work on learning embeddings of purely temporal processes expressed by ordinary differential equations. We turned to, again, following the qualitative theme, the learning of topological equivalence classes in ordinary differential equations. And today we're going to be talking about not ODEs, but PDEs, particularly reaction diffusion equations. And we're going to be learning or building representations of them that make their qualitative dynamics hopefully pop out by mapping them to some nice finite dimensional space. And that's because the key idea, I can move this, here we go, that's better for me. I'm moving my Zoom thing out of the way. The key idea or the key notion that we're going to be pursuing is that of the measurement. And that means that we're going to imagine sort of placing theoretically a sensor into the bath of the chemicals as they evolve and collecting information from across the spatial extent. And if that measurement is designed correctly, it should pick up those qualitative features of interest that are macroscopic across the spatial extent and should put us on a path towards such a qualitative theory. Because it's, in a sense, easy to build these measurements that distinguish between dynamical regimes, but it's hard to figure out how they behave in time and interact with parameters. So my cartoon here, which is sort of our goal in this, again, early work, is that we want to take some high, in particular, infinite dimensional family of partial differential equations, and their solutions sweep out some manifold over different parameters and initial conditions. I want to take a measurement of those systems that's macroscopic in its scope. And the output of our process is supposed to be a low, in particular, finite dimensional, now ordinary, that's purely temporal dynamics, which stands in as a surrogate for those high dimensional processes. And it's going to describe their qualitative dynamics. And the reason sort of we're focusing on reaction diffusion is because I've been outlining this taxonomy of patterns and regimes, which manifests in a very striking visual way. And that maps very nicely onto the notion of qualitative dynamics, because now we can start using visual terminology to describe their behavior in space. And so we're going to be making quasi-visual measurements and then figuring out how those behave in time. And this is based on recent work in the emerging field of data-driven dynamical systems, building on Koopman theory. Koopman theory is a way of building or approximating nonlinear dynamics with an infinite dimensional linear operator in measurement space. So now we're thinking about taking measurements of a system and not just relying on their states. Work from the Tokens embedding theorem, which is sort of about measurements. Other work in measurements about weak solutions to partial differential equations based on classical work by Galerkin. And the main benefit is supposed to be that we're going to directly model the macroscopic feature of interest without actually solving the partial differential equation. And again, as I said, the key is that it's easy to state one of these measurements. For example, a measurement that distinguishes between stripes from spots. But what's hard is to describe how it behaves in time and how it interacts with the parameters of the system. And that's going to be the...
Can I interrupt 1 second? I mean, just to be sure that I'm getting. From what I understand from this slide, it looks to me that you are essentially trying to do, let's say, a biological version of what we learn in statistical physics versus thermodynamics. I mean, you want to learn, what we learn in statistical physics versus thermodynamics is, for example, if you are interested in, I don't know, pressure, temperature, and this kind of macroscopic observable, you can give up the description of the microscopic dynamics and just describe, you can have a statistical description that is good enough to describe what you're interested in. Is it something similar here, that somehow you pick up the macroscopic observables that you are interested in and you want to develop a, let's say, a macroscopic description of these observables that somehow don't require the full knowledge of the full system microscopically?
I think the spirit is identical. I mean, and that's also just like that's like the story, as I understand it, of physics in the 19th century, sort of, at least as it applies to statistical physics. I think that one of the twists that we'll find is that the notion of measurement here, you offered these macroscopic measurements of temperature and pressure. The notion of measurement here is going to be more flexible.
So your goal is to finally describe the infinite dimensional family of PDEs, or you somehow give up the description because for your purposes only these low dimensional are interesting?
I think rather the latter than the former.
Okay. Okay, thanks.
Yeah, the latter rather than the former, and also, just from an engineering point of view, or from the point of view of thinking about the end user of the engineer, there will be some flexibility in what measurement means. In the sense that you can imagine, a thermometer picking up the macroscopic measurement of temperature, but it's more difficult to think about the sensor that you would need to pick up the macroscopic measurement of stripes versus spots if you were in the bath of the diffusing species. And so we're going to need to build in, frankly, some signal processing into the process.
Okay.
And that's the tweak. And our goal is sort of on the kind of a theoretical superstrate of the goals of the applied scientists, which is, first of all, system identification. We want to be able to make these measurements and then infer, in the sense of an inverse problem, the underlying parameters of the system. We're interested in control, and that's not just inferring the parameters, but then intervening and manipulating them so that the system evolves towards a target state, in the sense that its parameters actually now change. And then eventually we want to be able to develop a language with which we could communicate to practicing synthetic biologists who actually need to make these interventions, but often have to resort to these very exhaustive parameter sweeps. And that's the high-level overview. But now we can talk about some details of exactly how this is going to work, and then some early results and some things that excite us about the current approaches that we're applying, really in the last couple of weeks. So let's imagine that we have a family of solutions to a particular partial differential equation. Those solutions look like U of t, and we know that they evolve in time according to some equation of motion, which is in general the action of some nonlinear operator on U, parameterized by theta. And so this traces out some curve in an infinite dimensional space of, let's say, well-behaved functions. There's a lot of math, sort of bookkeeping to do, but you can think of them, they're just well-behaved in some bounded domain in, let's say, L2. Here we have an example of some initial condition evolving in time towards spots, okay? Then that looks like something you find on a lizard or something like that. And now what we want to do is we sort of want to forget about this high-dimensional stuff, which obscures the mechanism giving rise to spottiness. And we want to only compute some macroscopic measurement which picks it up, like feature detection. And again, as I said, we have some flexibility here. The simplest thing, which connects a little bit to what we were saying before about temperature or pressure, is just averaging, right? So you can just sum up the activity across space. Even this is actually, and not that easy to do, except in the simplest cases in an analytical way. You always, you tend to have to resort to approximative methods, even for this. We can get maybe a little more sophisticated, getting closer to the patterning notion. You could maybe look at the coefficient in the Fourier basis, and you can say, okay, what's how does this distribution of chemical species project onto a particular sinusoid, let's say? Or we could go one step further and look at time frequency localized features. So you take a little kernel, like a little wavelet, something like that, and then you convolve it with your distribution, and that gives you now heat map of where U looks like your kernel, and that you sum that activity up. And this idea can be iterated on in the matter of, frankly, computer vision. So there's flexibility here. But what we find in general is that it's macroscopic. We're taking averages, and we assume that phi as an operator is well behaved with respect to this solution, because what we really care about is the image of the solution in measurement space. So here we're working in infinite dimensions, but really the image of this solution in infinite dimensions under the action of the operator is some finite dimensional curve, frankly. And we assume that this is nice enough so that this curve is well behaved as a curve in a finite dimensional vector space, differentiable, so that we assume that there is actually some equation of motion here, which is simply obscure to us. And that's what we would want to know. We know it's differentiable, we know it's some low dimensional thing, and we want to understand how it behaves in time, and also in relation to those underlying parameters. Because remember, those are what the applied scientists are going to want to intervene at the level of. That's what they have access to. So, but we don't know how this thing behaves as a function of time, nor of those parameters. But we do know that there's an image that sort of lives above this high-dimensional thing in some low-dimensional measurement space, tracing out a curve that we'll call A. And so we don't know how A behaves, but what we can do, based on recent work in data-driven dynamical systems, is we can build a surrogate model that stands in not only for those high-dimensional dynamics, but also approximates the low-dimensional measurement as it evolves in time. And that regressed model we'll call a surrogate. That's a term from originally statistics. It's found home in physics as well. And the basic idea is that we don't know how this thing behaves, but we know that it's got some equation of motion, so we're just going to build it. And we have some options about how we can build it, but we'll say that the equation of motion is some function g, which is parameterized, and those are the parameters onto which we will regress from a data set. And crucially, it also depends on the underlying parameters of the observable partial differential equation. And what we're going to do is we're going to just adapt those parameters iteratively so that the estimated dynamics, according to the surrogate, resemble the image of the solution in measurement space. And we do so by comparing those curves in time. So we integrate up how they behave in time. And as a function of time, maybe we really only care about the beginning or the end or something like that. We want to figure out how the approximate solution, subject to the regressed parameters theta, behave, compared to the true image of the solution in measurement space, which depends on the true parameters. Note here, and we can talk about this in more detail, it's a subtlety, that there's a sense of learned parameters. Those are the regressed parameters, which I'm calling pi. And then the true parameters are the physical parameters of the system. But I'm using them with the same sort of notation here. But the idea is I have a model, I have an image of my system, kind of shadow of my system in measurement space, and I want them to be close. I do so by comparing them in time. And that regression can be symbolic or neural. By symbolic, I mean we can express G in a function dictionary. We can say G is Taylor series, G is Chebyshev polynomials, or whatever you'd like. And we can learn the coefficients there. And then with techniques from sparse symbolic regression, we can actually get a closed form on A, which is nice if you want to know what the functional form of your low-dimensional dynamics is. Or we can use neural regression. And by that, I mean, now we'll construe G as a neural network, those things that have sort of taken over our lives. And we'll regress onto the hidden parameters of that neural network. Both are possible and have their costs and benefits. And the data that we use for regression consists of the original high-dimensional solutions over many parameter settings and over many initial conditions, construed as images in this low-dimensional measurement space. We're going to think about the data as kind of sweeping out over parameters and initial conditions. And I'll just re-emphasize that this surrogate model, this surrogate equation of motion, shares a parameter space with the original nonlinear operator of the partial differential equation. And that's crucial because now, in order to make interventions at the level of the quality of the macroscopic feature of interest, we need only intervene at the level of the surrogate, since it's expressed in the same parameter space. And by now, this gesture of regressing a physical model in data is a standard method, including one that I've pursued in earlier work at the towards the ends of, again, optimal control and for symbolic regression, some work we did on reconstruction of governing equations. So this is now become a standard method in particularly data-driven dynamical systems. And that's the basic approach. And what we'll talk about now is an application where this measurement idea and the notion of regressing onto these low-dimensional measurements finds a simple expression. We'll talk about a couple of early results and then some of the stuff we're thinking of for the future.
So what is L? L is some length function or what?
So L is, excuse me, I should have said this. L is the criterion. It's a scalar positive value loss function, something just like pointwise Euclidean distance,
Okay.
something like that.
Okay,
And
so-
often I include this in as a function of time simply because, very often we don't care about transients. So there can be a cutoff here, or we'll say we only look at the last time step, etc. L is a loss function, objective function, which is positive value. Maybe it's convex too, if you're lucky. Okay, so let's talk about a very simple application. The application is going to be first a synthetic data from the Gray-Scott model. That's a particular reaction diffusion equation, which you'd encounter as a pattern generating system with certain biological correspondence. I'll say correspondence to biology. It has the following functional form, and it's a good example of a two-species reaction diffusion model taking the form of an activator-inhibitor system. And so by activator-inhibitor, we mean that the species U is a slow diffusing species, which is autocatalytic. It stimulates its own production, but it moves slowly in space. V, on the other hand, has an inhibitory function. It diffuses fast, and it inhibits its own production and the production of U. And in many cases, either one species, U or V, the activator or the inhibitor, will sort of win in the contest of dominance in the bath of the chemical species. But there are parameter settings where as a function of F and K in particular, less so the diffusion constants, where you will get an intermediate condition where now you have spatially heterogeneous distributions of U and V as a steady state, and those are what we colloquially call patterns. And so the author Munafo showed us how we can visualize those as a function of K and F. And we see that for large regions of parameter space, we can talk about how he made this visualization. It's kind of interesting. You get spatial homogeneity. So this is all U or this is all V. But along this sort of crescent-shaped bifurcation frontier between these two dominant regimes, you get this whole zoo of interesting patterns, ranging from, as I said before, that famous stripes to spots transition, but really everything in between. You get these bubbles, you get all sorts of labyrinthine structures, and then at the temporal level, you don't just get steady state stuff either, and this is something we haven't explored exactly yet, but you get chaos and solitons and other esoteric dynamical regimes. And the reason that this is nice is because it gives us a lot to look at, gives us a lot to, if you will, measure and see how those different things evolve in time as a function of those parameters. And the reason you get this beautiful...
Sorry, these funny shapes are not in space, right? They are in the space of F and K.
Yes, although Munafo, clever Munafo, made both, made the two coincide. So what the way he produced this figure was by solving the Gray-Scott equation numerically, but using such slow diffusion so that locally patterns would form in these at this scale. And before homogeneity could take over the whole spatial plane, he stopped it. And by this, I mean that F and K, according to Manaflo's visualization scheme, are actually varying in space. And that's how he made this figure.
Ah, I see. Okay. So should I take this as a sort of how it look like in space in a certain set?
You could look, I would think of it as more or less just like a library of images. And that if you wanted to see, in fact, he has a website that you can do this with. If you want to see how the behavior behaves at K equals this and F equals that, you would click on this thing and it would give you stripes, something like that. It just so happens that they're arranged like this. He took advantage of the fact that it's a two-dimensional spatial model with two parameters, basically.
Okay.
But in so doing, made this beautiful figure, which I rely on heavily. And the reason we get all these interesting things is because, again, you have that diffusion-driven instability, which we can talk about, which is now called a Turing instability after its discoverer, Alan Turing, who wrote about it amazingly in his last paper in 1952. So this is where we're going to start taking measurements and then learning how those measurements evolve in time. So what's the simplest possible measurement we can work with? Well, it's basic averaging. So now what we're going to do is we're going to consider a simple averaging measurement. And what that does is it takes in both species, right, which are now you can think of as, these are distributions over space. So these functions, infinite dimensional things, and the operator is going to report a two-dimensional signal. That two-dimensional signal is the concentration in U and the concentration in V. And we can observe at the outset that on the long term, that the measurement, which we denote by A, tends towards 0, 1, so no activator, all inhibitor in the green region, and the opposite in the red region. And that's precisely because of the activator inhibitor dynamics and precisely because of what we saw here in these two spatially homogeneous parameter conditions, right? But this reports essentially the coarsest possible thing, temperature, if you will, of gray Scott. But we don't know how it behaves in time, and we don't know how it behaves as a function of those crucial parameters, F and K. We can make measurements and we can observe it, but we want to build a model of it. In particular, we want to build a model that can learn how it works over here and generalize to how it works over there. That's sort of the magic of the data approach. And again, just for the simplest possible sort of sanity check condition of averaging. And so what we're going to do to start is we're going to learn a neural surrogate. So that means that this is going to be a neural network. We can talk about those details later, how that works, but it's it does take there's sort of theory to the way the construction of these things should work that needs to be deployed in order to have this function successfully. But for now, you can think of it as simply as a model to which we can regress certain parameters. And there's that loss function occurring again. And we're going to train this system using this loss function, which sort of looks scary, but it has a basic form. Namely, we're going to compare the estimated and the true measurement dynamics in time. And we're going to do so by really only looking at one time, the long term at big T. We're going to let it run for a little while. This is a Dirac delta, so we're going to kill everything else. And in particular, we want it to be close. We want the estimate and the true to be close in the sense of state. So this is the state in measurement space. And we also want it to be close in the sense of velocity, because we know that not only does the averaging operator tend towards particular states, here colored in green and red, but it tends towards steady state as well. And we want our surrogate to not just seek out those states and have a random velocity, but to also respect the purely dynamical aspect of its being at equilibrium at that time. So we want to match it in position and velocity at some large t. It's another way of saying it. And we're going to, again, train on numerous theta. Here, theta comprises F and K, and different initial conditions as well. Because we want our surrogate to really function like a flow. The flow maps from an initial condition through time to a final condition as a function of some parameters, and we want it to function that way and to capture those essential measurements in time. And then we're going to test it on some theta and initial conditions, which we held out. We wanted to generalize its knowledge. And so this is the simple, of course.
So I understand how you match the parameters theta between A and B because, well, they share the same parameter space, but how do you match U0? How do you match the initial condition?
So the initial condition is simply set as the initial, the model is given the initial condition. So maybe the best way to do it is to actually go back here. Yeah, so this has an initial state, U of 0. That has an image in measurement space, A of 0, right? And the hidden flow of the measurement dynamics transports A of 0 to A of, let's say, big T, something like that. The learning process consists of seeding, if you will, the surrogate model with the true A of 0. But after that, it's on its own.
Okay, so the surrogate model starts as the image of the true model.
Exactly.
And then you run the dynamics and you compare the endpoints.
Yeah, and in general, before training, it just goes, anywhere it wants to. And we have some flexibility about sort of what's the, what key points we want to target within the dynamics. So, maybe we have a scaffold of T's that we care about, or really only long-term, or after a burn-in period, or something like that. Try to write this in a general way. But to answer your question, you seed the surrogate with a starting point, and then you leave it to its own devices. And so what we're now doing is considering simply this basic averaging operator. We've stated the train test framework that we're going to be using. And let's see just the basic early results from, frankly, 10 days ago. So the first thing is that the surrogate qualitatively matches the bifurcation boundary on test data. So what I've shown you before is exactly what we're viewing in the left panel. So this is the true averaging measurement A after a long time. It has that bifurcation front, the crescent shape that we've seen before, as a function of F and K. Now, what we find is that the surrogate model on test data that it's never seen before, that's different initial conditions and different parameters F and K, when it's seeded on those initial measurements, we find that indeed it recapitulates qualitatively that bifurcation front. You get more or less the exact relationship between the long-term value of the measurement and the behavior of the surrogate in long time. So that's an encouraging initial result for a simple measurement. And indeed, it's not just a qualitative match, the values are close. And in order to validate whether or not our framework of learning a surrogate model, a surrogate dynamical system, if you will, in measurement space is really a good thing to do, because if you really just care about predicting how a system behaves in the long term, there's a lot of ways of doing that. You can learn any sort of map or any sort of heuristic that you want to just, given an initial condition, sort of guess where it ends up after a long time. But suffice it to say that it's actually not so simple, and that the commitment to an ordinary differential equation that you can regress onto, that lives in that measurement space, is actually a good, if you will, inductive bias. We say that in learning parlance to mean it captures the essential structure of the system that we're trying to model, Because we find that if we actually abandon that and we just try to sort of map directly from initial condition to final state, that we can't do it at all. And that's what we see in the right panel. You'll notice that it's in black and white, but that's actually because we're really only viewing one of those channels. You remember that before it was a two-dimensional averaging function of U and V. Here we're just looking at U, but the V looks exactly the same. And you'll see that if you just try to map from the initial condition to the final condition, you really can't do it easily, which kind of confirms our suspicion that this is a good thing to be doing, that the dynamics in the latent space are real, and we should treat them as such, and then learn a model, physical model, that recapitulates their dynamics, to take the dynamics seriously.
Excuse me.
Yeah.
May I ask a question? Yeah. So
of course.
With this kind of averaging that you're looking at, aren't you just missing all the interesting part, which is on the transition, where you have all the activity of, patterns and so on and so forth? Because the green and the red is, as you said, basically homogeneous one species or homogeneous the other species. But the interesting part of the dynamics was in the interface, right? Between the two regions.
Oh, absolutely. Absolutely. And you will see in a couple of slides some early work about how that's going to work, about how we're going to treat that. What we're viewing here is, you have to kind of crawl before you walk, in a sense. And I'm very excited to show you how well I can crawl here by validating as a sort of sanity check the transition between the two coarsest regimes you could ever want. And it's true that there's a lot of magic hiding along this bifurcation frontier, and we're going to see how we can interact with it soon. And that will, I hope, demonstrate why the flexibility in our choice of measurement, or why we leave that as a sort of flexible option, is a good thing, because it will allow us to make a choice about what we want to observe. We can observe pressure or temperature. In the same way, we can observe spots or stripes or anything in between. But it's true. This is what we're going to be heading towards in the long term, hopefully midterm. Now, you remember that I didn't just want to fit the state. These are dynamical objects. They behave certain ways in time, and we want our surrogate to recapitulate them. And you'll recall that our loss was designed exactly with that in mind. It matches position and velocity. And so to observe whether or not the velocity was also captured, namely the equilibrium behavior in time, what we did was we took the surrogate model, and then we evaluated it along a line of parameters. This cyan line at k equals 0.05. And at each case, we let the model run, and we observed not only its final state, but also what it was doing when it got there. Is it at a fixed point, or is it, blowing up to infinity, or doing something else that we really don't like? Because at that point, we would not be modeling dynamics. We would just be learning a map or building an input-output machine, which is not what we want to do. And what we find is that as you transit up this line at these steps, you move from f equals 0 to 0.11, and you move down these lines here. And we find that when you initialize the system, not only does it move to the proper states, you see that here, the red line stays red, or it stays at 1, because that's an activator-dominated regime. And as you move closer to this boundary, you see that it gets a little bit weaker, and then once you're in the interior, it terminates at this low steady state. But also, indeed, it is a steady state. These are flat lines. When it arrives at the long-term state, it's stationary. Okay, so that means that we're learning something about dynamics. We're not just learning an input-output machine. And that means we can then use our surrogate to ask, presumably, dynamical questions about how these macroscopic quantities of interest behave in time, and not just at a certain point, what is their state? Okay, so we took that as a nice secondary sanity check. And as a sort of final sanity check, I mentioned earlier that we're interested in control. So we want to intervene in these systems. And what we find is that the surrogate accelerates the control problem. And to that end, what we did was we look now at a single channel. Let's look at the U channel. We know that in the interior of the bifurcation region, it goes to 0 or some low value. And outside it goes to 1 or some high value. But we might want to influence that system. Well, how do we do it? Well, what we can do is that we can just take some initial parameter values in the interior, and then we can ask the question, okay, well, what's the nearest parameter such that the obverse behavior obtains? And by that I mean that the concentration of U doesn't go to 0 or some low value, it actually is maximal. So we want to find the nearest point in parameter space such that the opposite behavior obtains. And what we find is that by performing a sensitivity analysis, by figuring out how F and K relate to that macroscopic observable on the surrogate now, not on the full PDE, that we can indeed achieve that control problem, or we can solve that control problem, and we can do it much more quickly. From a numerical point of view, and from the point of view of applied scientists who want to be using these tools, that's one of our secondary goals. And what we find is that we get an enormous speed up on this test data compared to actually solving the PDE. Because you could solve this in the observable space. You solve the whole high-dimensional thing, and then you make the measurement and then compute the sensitivity. But we find that you don't need to do that here because you have access to the surrogate, which sort of validates its representational quality. It stands in, it's a surrogate for the essential dynamical elements according to a particular measurement which we chose. So this shows that, okay, maybe one of our goals of control, intervention in the system, is achievable with this method, at least for the simplest possible measurement. But as one of the audience members astutely observed, this is indeed the simplest measurement. So what's next, or what are we doing right now? Well, we're moving on to several, I hope, interesting things. And the first of them is more sophisticated measurements. So here's this famous figure of Munafo that we've already looked at. We've already stated that the blue and the red are the boring regimes, and the action is going on at this frontier, and there's a lot of things happening. The question is, okay, what measurement dynamics do we want to learn? And I'm sort of inspired by my eyes, and so that means, okay, I want to be extracting visual features, and that's a sort of colloquialism. But if we want to talk about it in a more rigorous sense, we could talk about taking our solutions, these distributions of these chemical species, and looking at their representations in some time frequency localized basis of wavelets, basically. So you have these little kernels that pick out little chirp-like features, and they can be parameterized by orientation and phase and scale, and convolving your distribution with them, in a sense, picks out those features in the form of a heat map, and then summing them up says how active is that feature in the image. And what this allows us to do with the proper choice of these filters, high pass filters, you can think of them, we can get nice coefficients, A, which can now give us access little by little to those interesting things happening at the bifurcation boundary. So here's an example where we took a bank of 3 Gabor wavelets, and we computed the representation of the steady state dynamics, or the steady state distribution of species for the Gray-Scott model with those 3 wavelets, or those 3 filters. You'll have to forgive some missing data, which is represented here by the white points. But what you can see is that, okay, now for the first time, we've got access not just to the boring zones, but to something going on at the bifurcation boundary, right? We now see a distinguished zone in red and a distinguished zone in pink. And I can sort of tell you that by from visual inspection and correspondence between these two figures, this is stripes and this is spots and this is stripes. And now the question is, how does this evolve in time? So that's one of the things we're looking at. And just by empirical observation, we can start looking at how those coefficients A, B behave in time for synthetic data on gray sky by observing, this is the true measurement data, A, as it evolves in time. So we have the trace of one measurement in time, A1 in red, and similarly for A2 and A3. And then we can actually play the same game for now real data, because that's our, we think of our end user as wanting to bring to bear some theoretical tool for the purposes of understanding real data. And sort of preempting the applied scientists, we're starting to look at how these measurements would behave on a real system. So earlier I showed you in one of the panels this evolving protein dynamics called the min-DE system. Min-D and min-E are two types of proteins. And we can observe how they behave by fluorescing the different proteins. And now what we can actually do is we can look at real videos of those proteins evolving in time, and now start to observe them behaving in measurement space. And the question we want to start asking is, okay, now that we have access to these more sophisticated measurements, and we have access to a theoretical apparatus which will allow us to regress a surrogate onto those low-dimensional dynamics, and presumably give us access to their dynamical behavior, what type of questions can we start asking that have biological import?
So
yeah.
We want to have time for questions, so maybe, I don't know, a few more minutes and then we can.
Of course. So, and I'll just say, this is where we're at. This is what we want to be doing. And it's something that excites us greatly and gets to the question of one of the audience members. And I'll say, thank you for your time, and also to my exceptional colleagues who I've noted here, and all of our funders. So I'm very sorry for going a little bit over, but I was happy to engage with your questions also during the talk. So at this point, I'll just say, I'm excited to hear, to have a discussion with you.
Okay, thanks very much.
Of course.
We can start with questions. I just had a question and then we'll open for other people's questions. So you had this L function which you defined, and you had A minus B and A dot minus B dot.
Yeah.
But is T supposed to be some, I just don't understand why A dot and B dot should be allowed to vary if you're going to just look at a specific T. Don't you want A dot and B dot to go to 0? Otherwise, it's not really an endpoint, right?
Well, I mean, in general, A dot and B dot, I mean, A dot is the true dynamics. It does whatever it wants. In most cases, for the patterning systems, it will be 0, because they're steady state. But B dot, which I haven't necessarily notated correctly here, is the behavior of the surrogate. Before training it can achieve the target B and still have the incorrect velocity. And in general, if you don't encourage it to do that, it won't find it. So
I don't understand why they should be. You want them to coincide at exactly small t equals capital T? That doesn't seem to make sense. I mean, if B dot is varying, then if they're equal at small, if A and B are equal at small t equals capital T, they won't be equal, for example, at a little bit later. So I just don't understand how.
Well, I mean, I'll first say that this is sort of, I hope, it's probably clear, but this is my shorthand for saying that, everything else is 0, right? So outside of this, we don't care. So you can just think about this whole function as being evaluated at big T.
Sorry, capital T is supposed to be an endpoint, right?
Yes, in this case it is.
So you shouldn't take it to be too early, otherwise it doesn't make sense. to be.
Oh yeah, absolutely. T is big. We can think of this T is big, and what delta is doing is simply just killing this function, except when T equals big T.
No, that I
So this is
understand.
evaluated at a single point. Yeah.
That I understand, but if A dot and B dot are not zero, I just don't understand why you should think of capital T as an endpoint. Because the things are still varying, so it shouldn't be an end, so it's not an endpoint.
Well, in the case that we're looking at, it is an endpoint, and we want them to achieve the appropriate zero velocity at the endpoint, being in equilibrium. But there's a lot going on in these systems which is not at equilibrium, and I sort of want to leave open the framework for the possibility that there are non-stationary behaviors which we also want to model. And so that's why I'm saying in general that our loss can be constructed in such a way that we needn't, we can maybe try to fit transients if we really want, and we don't even need to kill the rest of the solution. I'm just trying to say that there's more temporal machinery we can bring to bear here. I hope that was clear from this. But in general, your description is exactly correct.
Yeah, to follow up on this, I'm actually very surprised that you don't care about the transients. If you want the time scales of the dynamics to be fitted, that's one fork in the stationary state, right? Because if A dot equals 0 and B dot equals 0, that doesn't tell you that A and B will converge at the same speed. So why not include the transient? Why don't you care about the transient? It sounds like it would be more informative.
It would be more informative in the sense that it gives you a sense of the scale of the approach to, like, the time scale.
Yes.
I think that's an excellent direction to pursue, and I think that fitting that is an interesting next step. And in general, what you could do is, when I express the loss function, in general, I'm thinking about fitting the curves in their entirety. So regressing onto these solutions exactly, right? But that can be hard and implementationally slow. And so from a purely self-interested early stages of research, I opted for steady state, especially also because when you're communicating to biologists, they can be interested in transients and timescales from the developmental perspective, and especially the synthetic biologists working in protein dynamics. That's true. But oftentimes we're thinking of patterns as stationary objects which appear after a long time, and we can sort of forget what happens in the meanwhile. So that was sort of my informal justification for starting from this. But I mean, I'd be fascinated to start thinking about transients and matching timescales too, which is sort of why I tried to frame it as generally as I could.
Sorry, related to this, I don't know if what I'm gonna say, let's say, will break the rule of the game. Let's say I'm a bit confused on what you can assume that you know and what you cannot assume that you already know. So, for example, talking about this velocity constraint that you are inserting, what about putting an integrated in time, matching integrated in time this difference in the velocity, which maybe would be more relevant. Like from time to time, you match that the A and B dot are close to each other. Would it break the rule of the game in the sense that you are using an information that you're not supposed to know? Or
no, I don't think so. What we assume we know is the true measurement in time. We assume that the real system is in the other room, the full high dimensional differential PD is in the other room, and we're listening through the wall, and we only get a 3-dimensional signal out or a 1-dimensional signal out, but we've got the whole thing. And your description of matching velocities in the transient regimes, or before the endpoint, or at several other points in the interim, is, as far as I'm concerned, fair game. And I think it will also be interesting. And there's sort of also, numerical and data scientific questions to ask about how much information how little information can you get away with, or when you add more information, how does the quality of the surrogate change? And that's to say nothing of the difference between the neural surrogate, which is the one I've been working with, versus the symbolic surrogate, which would be especially interesting to start working with, because it gives you a closed form solution. But everything you said is fair game, and I'd be interested to pursue it.
Related, I mean, again, how much it's easy or hard? I mean, how you can decide what are the right measurements for a given model? I mean, you are at a certain moment, I mean, we discussed that this figure that you are showing now was not, let's say, particularly exciting because was missing J-series part. And then you said, okay, let's say, I can probably, let's say, start to see some of the patterns by adding this extra filter that you showed in. So I'm wondering, suppose that I arrive with a completely, let's say, unknown, I mean, completely a system for which it's hard to get an insight and so on. How hard, it's not as hard essentially as describing the full system, deciding what are the measurements that are relevant.
Yes, so I have strategically chosen combinations of systems and measurements that work for this early stage. Yeah, but I get, it is an open question how difficult this game is going to be to play as our desires to fit more interesting things increases. I will say that as we start to look at more sophisticated things, well, I'll say something cheap. The cheap thing is that empirically, it doesn't seem to be too hard to pick features that give us that distinguish between the regimes of interest. But that's also not scientific. That's anecdotal. One thing I will say is that the reason I've cited Mala here is that there's a whole science to building representations of these, let's call them visual or the spatial objects that expresses them in a time frequency localized basis exactly, right? So the idea is that the information is in there. If you predict if you choose the right phi and you choose enough of them, the information is in there. And so, for example, Mala has this famous scattering transform, which is sort of iteration on this idea. And the reason I'm sort of I'm bringing this up is because I do have a certain sense of confidence that, given the expressive capabilities of these feature extraction objects, that there exist transformations like those of Mala, the scattering transform, for which you could simply deploy it, distinguish between the patterns, and forget about it. And that this hardwiring of measurements that I'm engaging with now is really just because I'm at the early stages. And that at a later stage, I'll be able to rely on the fact that there exist such bases, which should theoretically distinguish between all of these, and that I can kind of work with universal measurements. Or frankly, and this is the secret, start learning the measurements, right, in an end-to-end way. But that's another story.
Okay.
I have a question about the size of the parameter space. So in your training, I mean, in your test systems here, you matched the size of the parameter space of the surrogate model to the size of the initial parameter space. But experimentally, how do you know that you have all the parameters that you need? How do you know that how can you match the parameter spaces since you never control all parameters? And for instance, in your min D, min E system?
Uh-uh.
In practice, what would you do?
Yeah. I sort of sense two questions, but maybe one of them is just my initial misunderstanding, and then you can correct me. In the regular framework for test and train and test, I play the regular machine learning game, which is I generate some random data uniformly over initial conditions and parameter space. I just kind of shotgun distribution, and then I split it, right? And there's the classic questions that we can ask from the data scientific point of view about, okay, am I interpolating or am I really extrapolating out of distribution and things like that? And those are interesting questions to pursue. And that's sort of an initial response to what I interpreted as the first part of your question. Now, the second part of your question about the parameters as they relate to real data, I think has some subtleties hiding in it, because the true model for this min-DE protein system is unknown. There are, of course, detailed biophysical models of how these things behave, but they're very different from the way Gray-Scott behaves. Now, underlying my juxtaposition of the two figures is my supposition that even though the parameter spaces are vastly different, we should be able to align, in a sense, a given real model from, let's say, min proteins, which has unknown dynamics, align that with a simple synthetic model, and that according to the surrogate, this system is, let's say, localized in the Gray-Scott sense at a particular F and K value, because that's all the surrogate knows. The surrogate knows how F and K behaves with relation to the measurement dynamics. It doesn't know about anything else. But because it has access to measurements which look the same, and it has to sort of situate them in a particular, FK arrangement, it will make a best guess, or at least you can compute in the sense of sensitivity which parameters best explain this. Now that's sort of limited because now we can't communicate to the synthetic biologists and say, well, in order to intervene, you need to manipulate such and such parameters, because now we're working at parameters that are parameter spaces which are different. And the response to that is that it could be, and this is something that we can develop in the future, that instead of working with a highly, specialized parameter space, these F and K, which tightly control known dynamical qualities or quantities in, let's say, the Gray-Scott model, we can now work with let's say, a function basis. And this is the game that they play in data-driven dynamical systems. So now we're going to be expressing the dynamics as a weighted combination of, let's say, polynomial terms. And now we'll play the same training game, not just over Gray-Scott, but over just arbitrary synthetic reaction-diffusion data as it emerges as random linear combinations of those basis functions, right? Constructing that data set is a little tricky, but let's assume we can do it. Now we can rest assured, just by grounds of function approximation, that our surrogate can localize an actual model for the protein dynamics, simply because it has access to a highly expressive basis of function. This connects to some of the early work I did a couple of years ago on reconstructing governing equations. Now that's like saying what the time is, and I told you how to build a watch. But suffice it to say that this is the subject of like very intense ongoing discussions in our group now. So I had a lot to say.
Rogerio, you had a question?
Yeah, I have 3 short questions. The first one is that the loss function is arbitrary. So did you try other loss functions? The second question is about parameters. So you have the Ricci parameters and you have the parameters from a surrogate. So the number of parameters have to be the same. They're the same type of parameters. And finally, the third question is, suppose you're given the result of a system which you don't know the equations. You don't know the parameters. Can you apply this method?
Yeah, okay. So let's start from the beginning. So the loss function is arbitrary. Yeah. Have I tried anything else? Yes, I've tried two other things. The first thing I tried is forgetting about the derivative. And the solution there is you get this, which is matching in state, but you don't get this. You see that these, the red curve will, approach the steady state, or what should be the steady state, at this oblique angle. And then you let it run for a little while and it blows up, or it doesn't represent the way the function behaves in time. I've also tried regressing onto the entire curve. That also works and reproduces the behavior that I've tried to model here, but it's just much slower. So in the interest of capturing exactly what I wanted and nothing else, I asked it to do only that. And as you can imagine, there's a million other things you could do. You can think about you can think about it as a function in time, and you can, maybe match spectral components of the curve. Somebody mentioned transients and matching timescales, all sorts of interesting things, but I haven't done anything like that yet. And I want to. The second thing you said was about parameters, neural parameters versus observable parameters. That gets to one of the subtleties here, which is that the neural parameters, or let's call them the regression parameters, because it needn't be neural. Those have no physical meaning at all. They simply control the functional form of which is either accessible in the form of symbolic or inaccessible in the form of neural, of the equation of motion. They don't have physical meaning. Now, you'll notice here that nevertheless, I have a parameter in this equation, and indeed it's the same parameter as the one controlling the true dynamics. That's because, at least for the assumption in training, is that the parameters of the data is known. And in fact, I supply it to this function, this sort of forward map to the tangent space, to the velocity. So these things are apples and oranges. And for now, you can just sort of forget about pi and just think that g is a function that comes to adapt to the true dynamics depending on theta. And the last question is actually related to that, which is, what do you do when you don't know the true equation? And that sort of gets back to what I was just speaking about to the earlier questioner. The simplest framework, as I've described it, is, okay, you know the equation, which is somewhat productive, but has its limitations too, and you don't always do that. And of course, there's a huge body of literature on reconstructing governing equations from data, which I've time to time contributed to. But I did say that nonetheless, there is a way to adapt this. And the idea with the adaptation is that if you now, instead of thinking about fitting to a particular equation, which has a known simple form, you can now think about not just sweeping over theta and initial conditions, but you're now sweeping over functional forms also in some parameterized way. So, you say that your reaction terms are all polynomial, something like that. Maybe some of them have reaction diffusion dynamics and some of them don't. In fact, most of them won't. But you can curate such a data set, and now you're not just, you're in a sense working with all equations at once. And it sounds grandiose, but it has concrete realization. And in that sense, you wouldn't have to know the true equation, which is exactly why in the case of the min dynamics, you'd be able to situate them in this function basis that you're working with more abstractly. That's a lot to say, and so I'd be happy to speak offline.
Yeah. No, thanks. That was very clear. Thank you, Matt.
Okay. Yeah, of course. Further questions?
Not a question, but a comment. I find this very exciting.
Oh, cool. Oh, that's great. I find
this sounds actually pretty promising, and I understand that it's pretty early in the project, but I'm really intrigued by the idea, and I'd love to see how it develops.
Well, hey, you and me both. And that I'm glad to have somebody take interest. We're really excited, and there's a lot of theoretical work to be done here, and I hope that in the coming weeks and months we'll be able to pursue some of those avenues and report back and see what we find based on your comments, which have been very insightful.
Maybe a quick question. I mean, it's going to open a whole new avenue of questions.
Yeah.
But it looks stochastic in the last figure, in the Mindy case.
Yeah, yeah.
How would you go into that direction? Include stochasticity in this?
Well, the first thing to do as a person who doesn't work on stochastic partial differential equations is to ignore it. that's which I'm sort of already doing. But if the Black-Scholes equation has taught us anything, it's that ignoring the behaviors of stochastic partial differential equations can destroy the world economy. So I don't want to do that, but I'll just these real systems are stochastic, both in the sense of like intrinsic noise. Proteins are noisy objects, and there's measurement noise as well. The fluorescent dynamics has its own intrinsic noise or whatever, and the instruments have their noise. I would be fascinated to pursue that. I don't know exactly how it would theoretically change the framework that I've laid out. Besides just putting the word stochastic in front of everything, but in that sense, I would need to rely on experts in that area in the form of collaboration. I'm sure, I'd be thrilled to talk to people who have that background. So, and again, an interesting avenue for this early work.
We should discuss this offline.
Yeah, I'd be happy to. Absolutely.
Any other questions, anybody? Okay. So thanks very much, Matthew. So I think probably by early next week, we'll we have a few more interviews to do, and then I think we'll be able to say exactly. But I'm glad you're still interested, and we'll get back to you very quickly.
Okay, great. Thanks so much. It's really been a fascinating discussion.
Okay. Thanks very much, Matthew.
Thank you.
Bye. By email, any questions come up.
Absolutely.
Okay.
Bye. Thanks, everybody.